shown here, I'll be discussing some of our very recent discoveries using whole genome methods, as well as transcriptome-wide evidence that have been observed in humans and model organisms. And I'm also going to present towards the end an application of that integrated methods in work that I've been doing with John McGeary and a few other folks at Clemson to see how we can translate some of that evidence to identify potential medications to treat different substances of abuse and potentially other kind of psychiatric illnesses that I won't be talking about today. And so as Paul mentioned, I'm a behavior geneticist and I have a background in mostly quantitative and statistical genetics. And over the course of my KO1, one of the things that I did was try to expand that into thinking about computational and system-based methods. And so like many of you here today, I'm really interested in getting to understand, how addictive related behaviors emerge from this combination of individual differences across different omic features, as well as the environment. And what's not represented here is the interaction between genes and environments, because oftentimes we're not well powered to study those, but it's just as important in all of our models. And so over the years, the research in this area has shown that when we look at substance use disorders, what we often find is that they're under moderate to strong genetic influences, and heritability estimates typically range from anywhere from 50 to 70%, depending on the substance that you're looking at and the developmental period. But one of the things that I've come to recognize over time is that we're really all working towards a common goal. And that goal is to understand the consequences and liability to substance use. But we're all coming at it from using different lenses or methods and levels of analysis, such as molecular, behavioral, systems neuroscience, or genetics. And each of these pieces of information is actually critically important. Using population-based studies in humans, we can infer lists of genes, but using model organism and in vitro experiments, we've actually already begun to understand how DNA variation can cause differences in response to substances of abuse. And so the challenge is how to bring those pieces of information together so they can inform one another and get us the answers that we want more quickly. Now, when it comes to genomic studies, the general observations that we've made from GWASs is that with increasing sample sizes, we're actually beginning to detect signals not only more reliably, but also more often. And this is because with increasing statistical power, smaller SNP effect sizes may actually be detected. But it's just important for us to underscore that even with recent advances that we're beginning to make in studies like GSCAN and the Million Veterans Project, that the GWASs are going to find variants that are going to match what we would expect, but oftentimes what we're also seeing is that there are variants that fall in poorly characterized genes, or we get what we call sort of sub-threshold or nominal associations. And this is posed a significant problem when it comes to interpreting many of these signals, as we don't necessarily know whether or not they're showing up because of, they're being linked to similar regulatory elements. And so we need to find ways that we can inform these types of studies. So getting to where we are today in human GWAS has actually been the result of an enormous effort on the parts of major consortia and organizations. But one of the things I recognize that it took two very large samples, right? So over 200,000 veterans and more than 50,000 alcohol-dependent cases to identify genome-wide significant loci for alcohol. In the last 2 years, we've actually gone from having just one locus to having now over 20-odd loci just for drinking behaviors. Now, what's interesting from these studies is that we've also been able to confirm what we've been able to identify years earlier in model organism studies for the role of alcohol and aldehyde dehydrogenase variants for drinking behaviors. But one of the interesting things we're seeing is that these loci are just important across different ancestries, even though we're underpowered to generalize a lot of the findings in European populations to individuals of African, Asian, Latino, and Native American backgrounds. Now, we've also been able to confirm that the genetic effects that we see both on substance use and problems matches up with what we've actually initially saw in twin studies, where we were able to discern that there's only going to be a partial overlap between the genetics on consumption and problematic use. And so it's really been great to identify those patterns of association, but more importantly, what we're also seeing is that the comorbidity with other behaviors seems to be different across those two different aspects of the substance use liability spectrum. And that's demonstrated in the figure on the lower left-hand corner. So One of the challenges that we've been having with the genome-wide association studies is that we still don't fully understand what the functional effects are of the many risk variants that were, one, passing our thresholds for genome-wide significance, as I mentioned earlier, but also the nominal ones. And there are many variable explanations for that, right? One of them is power, which we anticipate will improve over time as we get more and more samples. But another one of those is the fact that we need to start moving towards more sequencing studies, or essentially using long sequence reads so we can capture different types of structural variation and more rare forms of variation, albeit they're important, but they not necessarily contribute to a large part of the variance of the trait. One thing that also come up is whether or not we're asking the right or wrong questions. As I mentioned earlier, one of the things that we recognize in the animal field is that we can interrogate different components of the liability to substance use behaviors more so than we can when studying humans because of ethical limitations, as well as challenges to recruiting individuals at different stages of their substance use trajectory. That being said, now the outcomes that we're able to study typically in humans are those that are limited to diagnostic classifications, such as cases and controls. And that has posed a problem for us in terms of the phenotypic heterogeneity, right? Oftentimes what we're seeing is that genetic polymorphisms can actually vary across the different symptom criteria that we're using, but also the combinations of symptom criteria and how we choose to score them can affect our ability to detect the genetic influences that we're trying to describe. And just to give you an example of that, right, our research has shown that each symptom tends to be influenced by both shared and non-shared genetic effects that are actually poorly captured by just diagnostic outcomes. So you see here in the figure on the right that we published a few years ago, it's a study looking at DSM-5 criteria for alcohol use disorder. What we see here is that we can come up with a latent score, which has a pretty substantial heritability in comparison to looking at alcohol use disorder diagnosis, which is a heritability of 14%. But even so, when we look at the score, the amount of variance that's actually explained relevant to the actual symptom is not largely consistent across symptoms. So the variance that's captured, genetic variance that's captured by the latent structure can be quite high for some symptoms like recurring use, but quite low for others like spending a great deal of time using or recovering from that particular behavior, and from a particular drug use behavior. And so there's more work that needs to be done to understand what are the best ways to, identify the phenotypes that we want to study for understanding risk using humans, but also thinking about what are the best phenotypes in model organisms that might help us to overcome some of these limitations. And that's one of the factors I think plays really well into taking an integrated approach or integrative strategy. And so I think the ways that we can begin to overcome these limitations for GWAS is to take a much broader stance to thinking about evidence that we can capture using different types of approaches. And that creates a certain kinds of challenges for us in terms of, what are the assumptions we need to make about different systems or different model organisms, because the sequences might not necessarily align in the same way across those species. But we can look at similar systems because we might expect them to have similar processes that are related to the behavior. In the same way, we can expect that the behaviors across species may actually be similar in a variety of ways, and if they do diverge, we can use those differences as well to inform the sets of genes that we want to look at later on downstream in our analyses. And so this is work that's been done both by my colleagues Jason Dubier and Elissa Chesler at Jackson Laboratories, but we also know there are other organizations that are doing similar work. So the genenetwork.org, for example, has a large repository of data that one can access also on other platforms and like GeneWeaver to begin to get at these different biological features and integrate them as a tool, using tools into our analyses. And so there's several reasons why I think integrating evidence across species is actually very important. I think first and foremost, it affords us the ability to rapidly locate and translate loci for substance use disorders. And as I mentioned earlier, that's going to come with a variety of limitations. First and foremost is that it's still unclear how best to integrate the evidence, as well as figuring out basically whether or not there are certain paradigms from microorganisms that best map onto certain human behaviors more so than others, and vice versa. And so as we begin to get more information across different species, across different types of tissues, we need to conduct more studies. So I see opportunities where we can figure out what are the best ways to weight different types of evidence. So for example, should I put more weight on transcriptomic evidence over epigenetic evidence, for example? Oops, was that a sneeze? Zoom time. Right, so all these questions I mentioned earlier is just opportunities of galore, I think, for all of us, both in terms of the kinds of research questions or grants that we can generate to begin to fill some of these gaps. And I think that rather than waiting, we can leverage the current evidence using meta-analytic approaches to begin to understand how combinations of variants, for example, how combinations of EQTLs can help us to, prioritize variants for analysis, whether in, model organisms by doing some kind of combinations of upregulation or downregulation of certain genes, or essentially by looking them to see whether or not that set is relevant in a human population. And as you can imagine, this is not entirely new, right? There's been a large growing number of individuals working on this work. Some of who are already on this call. Jason Bouvier, for example, Kristen McNagna, Jessica Salvatore, as well as Spencer Huggett, have all been, and other researchers have been conducting studies to integrate evidence from other forms of addiction studies into GWAS, right? And so there's a growing number of studies along this line. In Kristen McNagna's most recent work, one of the things she's been able to do is to integrate evidence across, integrate expression evidence from the prefrontal cortex, the nucleus accumbens, and the VTA to actually realize novel gene networks for ethanol response and alcohol dependence, right? And so one of the things that we're beginning to see is that there is a link, right, that exists between GWAS and the consequences of exposure that if we take expression evidence, we can begin to realize. So for me, I believe that genome-wide expression studies provides a way for us to begin to understand how drugs are bringing about effects or changes in brain. And if we can identify the networks and the biological mechanisms or through GWAS that might be changing how these gene networks are varying in their expression, we can actually leverage that information, aka EQTLs, to prioritize sets of genes or sets of loci for future studies, right? I think that's even better when we have a situation where we're not just looking at expression evidence that comes from a tissue in a state that's not been exposed to a drug. And I think this is one of the big benefits of model organism research, that we're able to take animals that are exposed or not exposed and compare that to different extent to humans who may be exposed to the drug in the same way, albeit though we know that the level of exposure might not be something that we can directly map one-to-one across the two different organisms. So at this point, I just wanted to move on to talking about several of the projects that we've been doing in collaboration with researchers at both Jackson Laboratories, as well as the Psychogenetics Consortium Substance Use Disorder Working Group most recently. And a lot of this work has been led by postdoctoral fellows across these two labs, as well as Dr. Jason Bouvier, who's been working with them quite closely to help build the networks that we interrogate in these analyses. And so as illustrated on the right, what we've been looking for is the intersection of omic data in human and model organism populations with the goal of identifying functionally regulatory elements and or these sets of genes that are influencing related behavioral outcomes across species. And so the first study I'd like to talk about was just recently published in Translational Psychiatry last month. Actually, uses an integrated framework to investigate how genes that are associated with nicotine use in model organisms also contribute to the genetic architecture of human tobacco consumption. And so in this study, we tested 3 hypotheses. The first one being that DNA variation, both in the set of genes or around the set of genes, right, actually contribute to the heritability of human tobacco consumption. The second was that the model organism genes that we identified would be enriched for genes that are associated with human tobacco consumption. This is a sort of a gene level analysis. And thirdly, that a polygenic score, so this is applying the genome-wide association findings, would actually, based on our overall model organism gene set, would actually predict tobacco consumption in an independent population of individuals. And these turned out to be kids from the National Study of Adolescent Health. Right? And so all the samples that we'll be talking about here were all of European ancestry. And so the first thing we did in this study was to use the GeneWeaver platform to identify genes that were associated with nicotine use in a variety of model organisms. In total, we had about 712 unique genes that were associated with nicotine use that were actually also conserved in humans. We collected data from 5 different microarray studies that were done across mice, rats, zebrafish, and these were studies that spanned different brain regions. So talk about the ventral tegmental area, the nucleus accumbens, for instance. And so as shown on the left, one of the things that you see is that not many of these genes actually replicated, and most of our genes actually came from actual single study that was done in both C3H and B6 mice, right? And so one of the things that we've been working on over the years is identifying, can we come up with different thresholds for determining a confidence about a line of evidence. And so one of the things you'll see later on in the next study I talk about is that we move this threshold up from just being, the evidence that we observe across these sort of genes. So here we're using over 700 genes. In the subsequent study, when we look at drinking, I described a study on drinking, we actually set a threshold of 2 or more, so it had to have replicated. And that actually plays a role as well into the level of enrichment that we see, but also our confidence in the findings and the relevance of the genes that we're studying. And so we tested the relevance of this sort of model organism smoking gene set using both the genes that comprise the set as well as flanking genetic variation. And we did this in a sample of individuals who participated in the UK Biobank study. This was roughly about 123,000 individuals who were all of European ancestry. And as you can see in the figure on the left, for each gene that we identified, we came up with a variety of buffer region. This is buffer regions around that gene. And this is because we were interested in understanding the effects of the surrounding regulatory elements. But rather than just pick a particular size, we wanted to actually interrogate the sensitivity of our findings based on the size of the buffer length. And so we did analyses using no buffer length, so just focusing on the variation within the genes themselves. And that's because we actually imputed a lot of the information that wasn't on the genotyping arrays from UK Biobank. And so our analyses used roughly about 6 million loci. And so we did analyses using combination of variation, forms of variation. We interrogated the variation within the gene region, a buffer region around said set of genes, as well as looking at it in the context of all other forms of variation around in the genome. And the importance of this is that we get a conditional analysis for an additional conditional set of associations between the markers of interest, but also the set of interests. Otherwise, it would be necessary we could have inflated estimates if we didn't do that. All right, so when we talk about enrichments, it's going to be in the context of all other forms of variation that was imputed or on the platform itself. Okay, and so to do the analysis, we use multi-component mixed linear models using raw genotyping data. I would not necessarily recommend this approach in the future to anyone. It's computationally very, very intensive. One of the graduate students that are on the line, Pamela Romero, was part of this effort. She's now at CU Boulder, and she was part of the effort where we actually generated a lot of these GRMs, and it initially takes months to do so. And in the next study I'll talk about, we actually use an alternative approach, which is LD score regression, to try to speed this up. But in short, because we had such a large sample, our goal really was to see whether or not we could detect many of the same variants that were identified in the Liu et al. Study, but also identify additional sources of variation using a much smaller samples. And so one of the things to note here is that we did our analysis in triplicate using threefold cross-validation, where we divided the sample of smokers into 3 independent sets of groups of individuals, so roughly about 41,000 groups of smokers each. And one of the interesting things about this is that since we knew the actual heritability of the cigarette smoke per day in the sample, we were able to set it up such that we were well powered to detect an effect size as small as 1/2 of 1% of the variance in cigarette smokes phenotypic variance in cigarette smoke per day. So power-wise, we knew that we could get pretty small effect sizes. And so what we actually found was that we had support for all our hypotheses, and as shown in the 2 figures on the right, the variants that were both in, which is in the green, and around the gene set, the model organism gene set of interest, which is the ones in the red, accounted for anywhere from 5 to 36% of the observed SNP heritability of cigarette smoke per day in this data set. And so on the left, we kind of outline what the variation, the heritabilities were in the actual data set. So you can see the total heritability anywhere from 7.6 to 9.5%, depending on the fold you're looking at. So each set here that you're looking at, let me just come over here and highlight. So we have you're really looking at this first group here, along with its surrounding buffer, all other variation in the genome that existed. And then here we're showing in relation to the total. And so these three sum up to give you the total. And basically we have replication of findings across the different folds that we conducted. More importantly, when we look at the sensitivity of our buffer region, so we found that we observed enrichment in the regions around the nicotine model organism gene set, but interestingly, it decays as we increase the size of the buffer, in such that we still find enrichment if we were to go all the way out, to a buffer size as large as 50 kb around each of our genes of interest. But the signal to noise ratio seems to definitely be going down, and so that tells us that most of our variants are enriched around the genes of interest, which kind of makes sense because you expect the regulatory elements to be very close by. And so we did some follow-up analyses to try to make better sense of this, because there's new technologies coming out that help us identify where regulatory elements might be for specific genes of interest. And so another thing that we did related to our second hypothesis was to collapse data across both gene level analysis approaches. So these is MAGMA, H-MAGMA, and X-multi-X-scan. And in doing so, we conducted a gene-based test across the entire sample, so all 123,000 individuals, and we identified 115 unique genes that were actually associated with cigarettes per day. And 10 of these were actually also present in our model organism gene set, and 8 of them actually came from a study that investigated chronic nicotine self-administration in 5 brain regions using 2 different strains of mice. So notably, when we compared these negative control, some negative control gene sets that we have related to locomotor behavior and rat sucrose behavior, we found that these model organism gene sets that we were prioritizing and studying were still more enriched, and more so enriched than these compared to our negative control gene sets or any kind of gene sets that we permuted from our data of similar size to our actual model organism gene set that we tested. So we're pretty confident that we are seeing real effects here, and that these sets of genes are important. Now, to see whether or not it actually contributed any at all to variation that we'd expect in the same phenotype in an independent sample, we took the effect size distribution that we observed in our UK biobank, and we got the actual weights for each of the SNPs, and we applied them using a block-based method to create a polygenic risk score in the AdHealth sample. When we did that, we stratified the effect of the polygenic risk score into 3 components, the same way we did it in the UK biobank. So we had a PRS, so we did the total PRS, and when we did fit that model, you can see model 2. Okay, yes, all variants account for variation in cigarettes per day smoking in AdHealth, great. But if we wanted to stratify it into the individual components, so the variation within the genes of interest as well as around them, so using the buffer where we know there was still really good enrichment, but sufficiently large, so we did 10 kb, we found that we actually did find evidence that variation within the protein coding gene set of these model organism genes that were orthogonal in humans actually explain variation in cigarettes per day. And it's not a whoppingly large amount, right? But it is statistically significant and counts for roughly about 0.06% of the variance. So the second study I want to talk about is one that kind of sort of built on the study I mentioned quite earlier, where we kind of presented back in presented in Boston back at BGA in, I think this was 2017, but Spencer Huggett and another undergraduate who was in my lab, Janani Sujantan, who's since moved on, worked with Jason Bouvier to get more information so we can look at how different model organism gene sets, which model organism gene sets best recapitulate human studies of alcohol abuse and alcohol dependence. And so for this study, we had hypothesized that mammalian alcohol use models would actually parallel neuromolecular output from human alcoholics. So these are individuals who were drinkers who would have neurotranscriptional profiles on them. And the second hypothesis we made was that neurotranscriptional profiles of alcoholism would actually demonstrate higher convergence on paradigms of alcohol exposure, as opposed to naive strains of alcohol use. So these are strains basically that were selected for a particular line, but for drinking, but not necessarily exposed. So this idea of exposure, the transcriptome driven by exposure versus genetic predisposition. And so, on the left here, what I show you guys are the different studies that went into this analysis. So thank you all for all your hard work. We used all your data. And we conducted a cross-species meta-analysis of all these transcriptome-wide studies on alcohol abuse or dependence. And what we see on the right, is that gene sets that were most enriched were those that actually came from mouse and rat studies, right? Albeit when you look at the table on the left, you see that many of these actually varied in terms of brain region and phenotype, right? And so I think coming from this is there's still a lot more work that needs to get done as we get more data to figure out which brain regions are still most relevant, and essentially which species might still be the one that's really driving the results. But when we compare the transcriptome findings, one of the things that we noticed is that exposure models, so these are mice, rats that were exposed, seem to be best at capturing human alcoholism related gene expression. And so here we're just really, we're comparing the dots across the different experimental paradigms. And so this is just showing you the Jacquard similarity between the different gene sets within humans, and these are the model organisms expression lists. And we see here is that the exposure mice had higher Jacquard similarity estimates. We also observed that several behavioral paradigms of the exposure models actually work best. So this is just a correlation or essentially Jacquard similarity across species and behaviors in tissues. And essentially what we found was that there is significant overlap between human alcohol use disorder with binge-like models of alcohol use in both mice and rats. And there's some evidence to suggest that chronic intermittent alcohol exposure was also significant, but it's really sort of just barely there, right? And so clearly one of the things that to take away from this is that, yes, there's significant enrichment for both smoking behaviors. So we're seeing that for sure. And we're seeing that similarly as well for drinking behaviors, and that some paradigms seem to do a really good job of showing consilience than certainly others. But what if you wanted to actually go ahead and actually test for enrichment, right? Using the GWAS evidence. And so this is some work that Alyssa, Jason, and myself have been doing with the PGC. I think we're all part of the PGC Substance Use Disorder Working Group, and it's been headed up by a postdoc in my lab, so Spencer Huggett, along with Emma Johnson and Alex Atum, who sort of been running this work. And this work that will I'm going to be presenting part of this work today. It also extends to smoking, but I won't be talking about that. Just talking about the drinking.
Hey, Rohan.
And so, yeah.
Can I ask a question before you start this this next 2B? Back on the last slide, where you were talking about the different models. I guess, so I think this is really, really interesting, and I think it's really cool. You guys haven't published the alcohol stuff yet, right? I saw the nicotine paper, but I haven't seen the alcohol paper yet.
No, this one's coming pretty soon.
Yeah, so I guess the thing that I would just maybe caution you a little bit on in terms of interpretation is that obviously these correlations that you're seeing between the different phenotypes and the human phenotypes is going to be highly dependent on the data that you have.
Yes.
And that's already going to be somewhat biased based on the choices that people made in terms of what phenotypes they studied and what brain regions they studied. So I guess I would just hesitate to say at this point necessarily that like binge model does better than another model. One thing you might think about doing, and I don't know exactly how you did this analysis, but Spencer can probably figure it out, is, you know, doing a more of a different kind of connection between like the phenotype that was tested in the mouse with a phenotype that might be a little bit more comparable from the human. Because all that, it looks like you're just using some measure that's called alcohol use disorder here.
Mm-hmm.
And so, you know, obviously in some cases you have better sort of endophenotypes or subphenotypes. So maybe one thing to consider in this analysis is whether there's a way to make those connections a little bit clearer.
Yes.
Just a thought.
I love your thought. Yeah,
Okay,
I think Alyssa. I'm sorry, I didn't have the chat open up earlier. I think Alyssa also
it's the
chimed in as well.
Yeah.
This is one I tried to make a point earlier. Is like there are tons of areas for opportunity, because one of the things we recognize doing this is that we need more data, and I think just like what we're seeing with GWAS, you know, in doing any of these kinds of approaches, a meta-analytic approach makes the most sense going forward. As we get more data, we integrate it into these pipelines. And I agree, they're probably not to really oversell or overinterpret, you know, the conclusions that one can make from what we have so far. And this was partly, so 2A was well in advance of what I'm about to show you next, as we started to actually think about this. And with the paper that we're getting ready to put out in archive, which Spencer will talk about at the upcoming Nanogenetics, Epigenetics Consortium meeting in more detail, does take some use phenotypes into account, right? But essentially, you know, what we want to begin to do, this is my thinking, is to start to come up with studies where we look across a wide array of different paradigms in human endophenotypes, right, as well as behavioral phenotypes, neurophenotypes, and so we can see which ones best align. And it's not necessarily that it's about best alignment, it's about the agglomerative knowledge that we can get across all of them, more so than one being better than the other. Yeah. Because ultimately what we want is to get a set of information and figure out which ones we might want to interpret in certain ways. Because at present, we might have associations or correlations between our human traits and these variants, but in order to fully begin to interpret them, right, and think about how we can leverage them downstream, whether it's in a medication or, you know, as part of some kind of therapy or therapy's efficacy, we need to understand how it's interacting. And we can't do that without any form of manipulation or experimentation. And as I said earlier, it's just not something we can do in a person. And if we're struggling now to make discoveries about how this works with just blatant discovery in terms of just GWAS brute force, the idea of a quasi-experimental approach to realizing SNP effects in humans is going to be several orders of magnitude even greater. So for me, this seems like a win-win in terms of, you know, there's a big curve that we need to round. You know, let's just cut into the road, the mountain to our left, and go, you know, go right through to get to the other side if we can meta-analyze across some of this information. But we have to do it smartly, that's what I'm saying. But I agree wholeheartedly, Marissa, sorry. And thanks, Alyssa, for chiming in. Yeah. So where was I? I make the point too that I think Marissa's comment also generalizes to the subsequent studies that I'm going to show next. So in this next study, we tested for enrichment in recent large GWAS, discovery GWAS on drinks per week, as well as problematic alcohol use. So expanding a little bit. So these are studies from the Liu et al. Paper in 2019 and the Zhou et al.'s paper in Nature Neuroscience on problematic alcohol use. And again, we're using GeneWeaver. We had acquired experimental model samples and human cell exposure gene sets for alcohol. And we use mice and rat samples from previous study that I talked about earlier, okay? And so for our alcoholic cell exposures, basically we're talking about exposure where it's either in vitro exposure to some kind of neuroblastoma or lymphoblastoid cells that we obtained, that were obtained from alcoholics. Okay, so it's the context that is relevant. So in this study, right, we also identified gene sets that could serve as sort of control paradigms, right? And these are paradigms that we thought were somewhat relevant to the behaviors, but not necessarily relevant to drinking behaviors, right? So think about things like sugar consumption be a good control behavior, or locomotor behavior, right? So those sort of controls. But also to get specificity, one of the things we also wanted to do was to look at how our gene sets might actually be performing for traits that were similar in heritability or maybe higher in heritability in comparison to the ones that we're studying, right? So we also looked at how our gene sets were enriched in conjunction with 14 other non-substance use GWAS traits, right? Including neurological traits, neuropsychiatric traits, anthropometric traits, cardiometabolic traits. So we're talking about things like, height, BMI, schizophrenia, Alzheimer's. We threw those in as well just to see if we have any evidence for some specificity to our findings. And so just sort of foreshadow, give you guys a showcase of what we'll be releasing later this month. For our analysis, we actually end up carrying forward about 800 genes that were observed in roughly at least 2 different alcohol studies. So compared to the smoking study earlier, where we had a threshold of just anything observed, now we're setting a higher bar. So it had to be observed in at least 2 or more studies. I didn't show you that sort of, histogram, but there are more than 4,000 genes that have been identified and associated with alcohol use behaviors, right, across different kinds of studies. And so, but only about 800 or so of them actually have been replicated. And so we fitted models using these 800 genes using LD-PRED, and we put a 100 kb buffer around them for each gene, and then we corrected for the multiple phenotypes that we actually tested. Okay, so on the left, what we show is that we had the most overlap of the alcohol rodent models with humans drink per week, which roughly captured about 1/3 of the observed SNP heritability. So we see SNP heritability of drinks per week being roughly 4% in that GWAS. And we get to roughly about 1/3 of that using that model organism, using that particular gene set. We also see that on the right, that effect sizes of these effects also indicate that the rodent alcohol gene set was actually significantly enriched for heritability for the human GWAS of drinks per week with an odds ratio of roughly 1.4. And what we had for other gene sets that we use as sort of a control are also relevant. So things like the keg addiction pathway genes, as well as non-substance use gene sets, so locomotor, sucrose gene sets, did not show any kind of significant enrichment. And so here we're sort of seeing some evidence for these rodent model genes being relevant. When we did our sensitivity analyses, it showed that the gene set that's actually enriched, the gene set enrichment actually increased as a function of trait heritability. This is something that's important to take into consideration going forward, right? So depending on the traits that are being analyzed and the heritability of said traits, one might actually see, higher levels of enrichment than you would expect by chance. But you really have to take that into consideration, how heritable that trait is. And so when we control for trait heritability, what we find is that the rodent alcohol gene set showed greater enrichment in compared to similarly heritable human traits that we wouldn't expect to be associated with, drinking behaviors. And so, for example, wearing glasses, what side of your head you use your phone on, things that are, heart disease, major depression, for example, right? We all consistently find that our enrichment signal is higher in our drinking gene set. And so the last study that I want to talk about today is one that we did in collaboration with folks from Clemson, so both Robert and Trudy McKay, I'm not sure if they're actually on the call, along with John McGarry, who's on our call, thank you, John, and another colleague of ours at Emory. I don't know why I'm blanking on her name, forgive me at the moment. John, help me out.
Justine Welsh.
Justine Welsh, thank you, John. Justine Welsh, who who helped us to sort of translate some of the findings using a drug discovery pipeline that we came up with in the lab. And this pipeline really just builds on the connectivity mapping evidence that exists at the Broad. And the goal here basically is to showcase how we can take this integrative evidence to rapidly screen potential medications for addiction. In particular, medications that might already be FDA approved, right, but might not actually be considered as of yet for study. And so in this study, I'm going to talk about, we correlated drug expression based on cells in a dish, essentially, with disease expression in humans and model organisms. And so we started with cocaine, partly because, what we've seen in recent years is that rate of cocaine overdoses has quadrupled, basically, and moreover, what we're actually seeing right now is that there are only about 20 pharmaceutical medications that are undergoing clinical trials for cocaine use disorder, in comparison to several hundred of repurposable drugs that could also be potentially used, but haven't necessarily been screened, as far as we know, for being relevant for treating our disease. Right? And so for this study, we used the connectivity mapping tool. And as I mentioned earlier, we correlated drug expression with disease expression. So we have evidence from human brain as well as model organism brains, the drug effects in a dish, basically, and we're going to basically be meta-analyzing across all of those evidence. So these analyses use cocaine use disorder expression from human brain samples. We have a total of about 71 human brains that went into this analysis, and as well as the drug exposure profiles from 830 odd FDA-approved medications, and as well as validation data that we were able to actually pull from studies that were done on mice and in actual neuronal cells. And so we take all that evidence and we apply it in a pipeline, basically, that incorporates an element of validation. For our validation, we use transcriptome-wide cocaine self-administration data and human neural cocaine exposure data as well. And so all the drugs that we identify get validated in silico, essentially, using that data. And then we had, for the mouse cocaine self-administrator, we used the same brain regions as the humans that was shown on the previous slide. And for the human neuronal cocaine exposure data, we evaluated different doses and time points that were associated with cocaine use as relevant. The last thing we did with our collaborators at Clemson was to follow up with the drugs that were most, or the drug that was most relevant to our disease of interest. And I'm going to sort of explain that a little bit more as we go along. And so we took this multi-level meta-regression approach in order to match and validate and benchmark roughly about 835 potential treatments, right? And the benefit of this is that it allows us to remain flexible in our design, or as well as the weighting of the relevant, how to put it, compounds based on the expression differences that we observe across, that were intersecting or consilient across model organisms and humans. And so basically, we end up with a flexible, powerful, and interpretable framework. From it, we can actually find treatments based on the RNA associations. So it's all benchmarking RNA associations with humans, right? While also accounting for the different complexity or layers of the data. Because for the drug, we can have different levels of exposure. Depending on the model organism data, we can have different tissues, and so on and so forth. And so from this analysis, we found about 16 treatments that actually significantly, we say, were counteracted, meet the RNA neurobiology that's associated with cocaine use disorder. And so, I'm not showing this here, but we also have evidence about compounds that might have gone the other direction as well. But for what we're following up on, we're looking at the relevant change, the change relevant to the controls in our human population. So do we see a counteraction to the new RNA profile? And so of these 16 compounds, ibrutinib, which is the second one at the top, actually stood out as having the most consistent evidence across the 3 brain regions that we tested in humans. So midbrain, hippocampus, and dorsolateral prefrontal cortex. And so those of you not familiar with ibrutinib, It's a Bruton's kinase inhibitor, and it actually stood out across all these regions. We do notice, though, however, that there are several other compounds as well, and we're in the process of working on those and screening them. So one of the things we wanted to do as well in this study was to contrast the findings for our drug discovery pipeline with that of current compounds that are actually being treated. Now, we weren't able to get our hands on all the compounds that are in treatment that are currently being tested, not being treated, but that are currently being evaluated. But what we find is that in comparison to the drugs that were available in the L1000 dataset, we found that many of those compounds did not exhibit the same profile as the 16 that we discovered, right? And so they don't seem to be tied in any way to the neuromolecular associations between human and cocaine use disorder. And so we thought that was quite interesting. And so we carried forward, these 16 compounds. So looking into them, we found that they actually may be associated with cocaine overdose as well as toxicity. But it's very limited evidence to suggest that any of these compounds actually were linked to any sort of behavioral traits related to cocaine use disorder. And so for our validation, we took these top compounds and we looked at them in the data that was available, and to see whether or not the effects of our potential CED treatments, these are the 16 treatments, persisted in these 2 preclinical models for cocaine self-administration. And essentially what we found was that of the 16 compounds, ibrutinib stood out again as being the one that was negatively associated with neuronal cocaine exposure, right? So that's what's shown down here on the bottom, okay? You can see here as well at sort of different time points in that study. And it was also trending towards significance to reverse gene expression, right, with mouse cocaine self-administration as well. And that p-value was actually, the adjusted p-value for that analysis was like 0.07 for anyone who's interested in the FDRs reported here. So of the other 16 compounds that we looked at in these 2 paradigms, none of them actually were significant. We also sort of dove back into the data that was available on the drug expression from the drug treatment tissue, from the L1000 database, basically looking at differential expression as a function of the compounds that were actually used. And what we also found looking into that data was that ibrutinib seems to be reversing the expression of genes that are involved in both dopaminergic as well as glutamatergic neurotransmission, right? And we also see evidence for various immediate early genes, right? Those that are tied to as well as those that are tied to intracellular signaling, right? And a cascade of genes. Go ahead. Oh, I think it's a baby. Lars, you have a question? Okay. So working with our Clemson collaborators, we actually were able to examine ibrutinib's effects as a potential therapeutic by looking at how it affected both seizure behavior and startle response behaviors in Drosophila. And so what you see here on the left was evidence that both male and female flies that consume ibrutinib with cocaine actually spent more time moving around, so percent time seizing, less time percent time seizing, than flies that actually only consumed cocaine. However, we see that only male flies that consumed ibrutinib and cocaine showed a significant decrease in the prevalence of seizures. On the right, what you're looking at is that male flies who were exposed to cocaine spent less time moving after being subjected to a mechanical startle. So, this is giving us evidence to suggest that it somewhat is protective, right? And we're also seeing that both male and female flies that consumed ibrutinib and cocaine spent more time moving than flies that only consumed cocaine overall. So we found that sort of really, quite suggestive of sort of a neuroprotective effect. And so we're continuing to work with ibrutinib and going forward. So just sort of to sum things up, I know I'm kind of over time. I think we've seen that scalable integration of experimental models with human addiction genetics can be somewhat challenging because of all the assumptions that we need to make, but I think it's quite informative. And I think sort of piggybacking on what Marissa mentioned earlier, and Alyssa kind of reinforced, is that we need more data, and as we get more data, we can begin to ask these questions about consilience and translate it in a way that is in a meta-analytic fashion that is conservative, but also helps us to move further along. Because in the study I showed you on smoking, we took 40,000 individuals and we were able to identify basically the same number of genes that it took us with 1.1 million individuals for smoking. And at the same time, we're also able to demonstrate the benefits of incorporating the model organism evidence. And so some novel paradigms that are coming out right now allow us to integrate this evidence a priori into our GWASs. So like there's Bayesian functional methods that you can use to adjust the significance level of certain variants. So if you're interested in just prioritizing more loci or more genes, you can certainly do that. But at the same time, I think it's important to understand why you're putting that information in there. I think the same issue that Marissa brought up is very relevant. What we put in affects what we get out. But it's better than just taking, I think, just our GWAS findings and just trying to justify it based on the evidence that we already have. I think we should take it all, look at all of it together. We are seeing that model species and behavioral paradigms do differ to some extent in their degree, but we need more work needs to be done to understand, their relative contributions to different aspects of human substance use and substance addiction. In the same way, mouse and rat models of tobacco and alcohol exposure we see that they might best map onto neurotranscriptional profiles for heavy users and drinkers. And that kind of makes sense. We expect that if we have models that are reflective of heavy exposure or heavy levels of use, then that should be highly correlated with heavy exposure models in humans, because we see a high correlation between level of exposure in humans with the severity of their substance use problems. Of course, we do know that potential conserved systems are a good place to target. So for these kinds of analyses, neurotransmissions we saw in the study I showed on cocaine, intracellular signaling, and synaptic plasticity might be good places to start. And a good place to start if you're interested in drug treatment, developing drugs or pursuing drugs for study might be the drugs that are already FDA approved. And we're more than willing to work with anyone interested in collaborating or sharing their data to begin to apply this platform, this pipeline to other kinds of traits. So we're currently beginning to work on opioid use and depression, for instance. And so what's next for our team, I think most of you are on this call, is, as mentioned earlier, expanding, but also beginning to understand how convergence across different animal paradigms plays a role in different types of substance use behaviors, whether it's alcohol or smoking or cocaine or opioids. But I think it's just as important to think about, how other species can inform these analyses. So we are moving on now to do work that involves monkeys and how that can actually inform some of the models I showed you earlier. We also looking into comparing and contrasting predictive utility of different paradigms. And again, we're relying on our collaborators, Jason and Alyssa are kind of key to a lot of this work. But again, the Gene Weaver resource is freely available to everyone, right? So as we develop these kind of gene sets, we try to make them accessible to everyone. So as you make a set, one of the things that you can do is you can get that set assigned a gene set ID, and so it can easily be followed up by anyone else. And so they make it quite easy. Working with them firsthand, I can say very easy to work with for anyone interested in doing so. So we are following up on specific gene variants as well that might underlie human RNA associations with disordered drug use. And one of the things we're working on right now is, looking at how ibrutinib might play a role in different models related to cocaine exposure in using rodents. And so anybody who's interested in collaborating, again, more than happy to touch base with you. And I'll just end there, and here are ways to contact me. Rohan at Palmer@Emory.edu. I'm also on Twitter. This is my office number. And if you just Google me as well, my personal number is out there for the whole world as well. And that's all I have.
Thanks, Dr. Palmer. I'm clapping on everyone's behalf. I I have a question, but I'm going to open it up to the group first and ask if anyone has any questions for Dr. Palmer. Okay, so I guess I'll ask my question then. And it's a general question, but it has to do with Dr. Ehringer's word of caution and Dr. Chesler's call for more data. I just want to double down on both of those things. But when I was watching that part of your talk, I was thinking, you know, if I'm a neurobiologist, I'm interested in studying, you know, neural circuits underlying alcoholism, I might be wondering what sort of animal model I might want to choose, you know, whether it's rats or mice, or whether it's IP injections of alcohol versus binge drinking versus 24-hour access. Like based on what you showed, I'm going to be looking for the model with the best construct validity. And that's sort of like the data that you showed sort of suggested that maybe I want to go with a binge drinking model. I guess my question is that short-sighted, or is that actually a goal? Are we trying to get as much data so that we can actually make judgments on which of the animal models that we're using to find treatments and understand the basic biology are going to be the most useful moving forward?
Yeah, I think the classifier that you used there, which is sort of more useful, I think we're not at the point where I think we can discern that at this right now. And I think what the evidence that we're getting so far demonstrates is that how we've invested thus far has been beneficial overall. We have learned, what we're learning from paradigm A to paradigm E, E or Z, is they're all relevant, and each of them is telling us a little bit more about how humans or how these animals are responding. And I think if we're looking, it could be the case that we look across all of this, and at the end of the day, they're all equal in their effects, right? It's like the opposite on the GWAS side, right? You do the GWAS and you're like, oh, my SNP effect sizes are the same. I should just give up, right? But that's not the case, right? It's the case that it's figuring out, okay, well, it's not all the variants that are relevant to the disease of interest, right? Some of them more so than others confer the greater risk, even though the effect sizes might look the same. So I would suggest that looking at this, that we continue to do it. I think what you might want to do at that point is maybe you don't just write a singular R1, you find another collaborator who might do it who has the skills to do it in a different paradigm along with you, and across all those grants, you paint the bigger picture. And so then what would come out of it would be this sort of meta analytic approach. I think Carla just chimed in as well, that different models provide different perspectives, yeah, and various levels of mechanism. And that's basically what I'm also saying here. We need to sort of acknowledge the biodiversity and the knowledge we get from each of those different models, and then leverage that in the best way possible downstream. I'm not advocating here that one particular design is much better than the other. I think history might tell that down the road, but as of now, everything is beneficial.
For the record, I agree with everything you just said and everything that everyone's been saying. I just wanted to make sure that we had that conversation and sort of play that devil's advocate. I agree that we're not there yet, and we're all looking forward to creating more work for you by generating data using a vast array of animal models. So I don't want to pretend like I'm looking for the perfect model, although maybe down the road, like you said, we will be able to zero in on things where we could spend more of our energy, but I agree we're not there yet.
Yeah.
Are there other questions for Dr. Palmer? You could either mute yourself or unmute yourself, or put something in the chat. Okay. Well, Alyssa just put a comment in the chat as she's signing off, and this is probably a good point for us to sign off as well. But just to reiterate for a third time, we need all of your data, especially Alyssa needs your data. Dr. Palmer needs your data. So make sure when you're doing genomic studies to keep all of these folks in mind that are on the call. And then I want to end by saying thank you to Dr. Palmer for a really great talk. I had high expectations and you exceeded those expectations. It was really fascinating work. I think you're doing really important work trying to do these and successfully doing this cross-species integration of data sets. This is really, in my opinion, what it's all about. And I'm glad that you're one of the folks doing that work. And I just want to say thank you for sharing it with us today, and look forward to more stuff coming from you in the future.
Oh, thank you. Thank you. Like I said, we couldn't be here without all of us having done this work so far. And I think I see it as a concerted effort.
I agree. So big group hug, and I'll see you all. I'll see you all next month.
Yes.